Hello all. In this video we are going to see about gibberellin signaling. So far we have seen about in the series of plant phytohormones, we have seen about definitions and physiological roles of gibberellin. In this video we are going to see about gibberellin signaling. When and how gibberellin functions are turned on and how gibberellin functions are turned off. This is what we are going to see about in this video. First of all, we will see what are the components of signaling. In the signaling pathway, to undergo, what are the components are needed? We need, first component is the hormone itself, that is gibberellin. Second component is soluble nuclear receptor protein. First component is gibberellin, component number 1. Second component is soluble nuclear receptor protein, that is GID1. In short, GID1, abbreviated as gibberellic acid insensitive dwarf 1. This is soluble nuclear receptor protein, name of the receptor. Second thing is DELA protein. That is third thing is DELA protein. Next thing is polyubiquitination process by SCF. Fourth thing, SCF. These are the major components of gibberellin signaling. Major components include gibberellic acid, the soluble nuclear receptor protein, that is GID1, DELA protein, SCF, polyubiquitination, 26S proteasome mediated degradation. These are the major components of gibberellin signaling pathway. This is the overview about gibberellin signaling. We will see in detail. First thing, gibberellin will bind to this receptor, soluble nuclear protein receptor, that is GID1. Gibberellic acid insensitive dwarf. Gibberellin acts as ligand, will bind to this receptor. This binding is controlled by DELA protein. It is a proteinaceous compound named DELA. Generally, what if a ligand binds to a receptor? A conformational change takes place. The same thing occurs here. A conformational change takes place if gibberellin attached with the receptor. It is associated with N-terminal domain. We have two terminals, that is C-terminal and N-terminal. In N-terminal domain of DELA protein, we have sequences like ASP, GLU, hope this is known, leucine and alanine sequences. Leucine, alanine, LUG and ASP. These are the amino acid sequences of N-terminal domain of DELA protein. This is again an important thing. What are the sequences in the N-terminal domain of DELA protein? These are the sequences. ASP, I repeat, ASP, ALU, leucine, alanine. These are the sequences. One point is clear, how this receptor bind with this ligand. Next thing, DELA protein. DELA protein may be, what are the various types? In rice we call it as SLR1. In Arabidopsis thaliana, we call it as GAI. What is SLR1? SLR1 is slender rice 1. GAI is gibberellic acid insensitive. These are the various names given to DELA protein in different plants. In rice plants, it is called as slender rice 1. In Arabidopsis plant, it is called as GAI. These are the various other names given in specific plants. Now we will see what are the functions of this DELA protein. This DELA protein will act as a repressor with regard to gibberellic acid signaling pathway. It acts as a transcriptional repressor. Repressor in the sense it inhibits the pathway. Now we will see what will happen in the presence of gibberellic acid and in the absence of gibberellic acid. In the absence of gibberellin, this inhibitor, this transcriptional repressor DELA protein, this DELA protein will main function of this is repressing, that is inhibiting the pathway. This DELA protein will inhibit all the process of gibberellin. We know about the functions of gibberellin like seed germination, stem elongation, cell division. So all these things will be inhibited when this DELA protein when comes to bind in case of absence of gibberellin. This DELA protein represses gibberellin dependent processes like seed germination, leaf expansion, stem elongation, flowering and seed development. So what will happen? In case of presence of gibberellin, we will see what will happen in case of presence. In case of absence, all the functions will be absent because DELA protein will come and bind and it will repress gibberellin. Gibberellic acid functions. Okay. What will happen in case of presence of gibberellin? Case of presence of gibberellin, that is gibberellic acid insensitive dwarf 1 induces polyubiquitinylation. Ubiquitinylation induces polyubiquitinylation of DELA protein. What is polyubiquitinylation? Is nothing but complete degradation. If DELA protein is completely degraded, then gibberellic acid functions will be expressed. Complete degradation is the meaning of polyubiquitinylation. This polyubiquitinylation occurs with the help of SCF. SCF is nothing but shabby. Okay. What is SCF? SCF is nothing but escapee 1 culin F box protein complex. We will be studying F complex, F1 box complex in case of auxin. In case of gibberellin, we will see escapee 1 culin F box complex. Escapee 1 culin F box complex that act as a ubiquitin ligase that catalyzes the reaction of polyubiquitinylation of DELA protein. And DELA protein will undergo polyubiquitinylation with the help of 26S proteasome. DELA protein will act as repressor and they may also act as an activator. The major thing is about presence or absence of gibberellin, and DELA protein will act accordingly. escapee 1 culin F box is an complete expansion of SCF. It acts as a ubiquitin ligase. Okay. So let's sum up. Gibberellin acid pathway, signaling pathway becomes very important for CSIR exam with regard to the functioning of DELA repressor It's asked multiple times in various competitive exams of life sciences. Okay. So we will see. The major function of DELA protein is transcriptional repressor. If gibberellic acid is present, what they will do? They will induce polyubiquitinylation mediated proteasomal degradation with the help of 26S proteasome. They are induced by ubiquitin ligase with the help of SCF complex. This is one side story. The another side story, if gibberellic acid is not present, DELA protein will come and bind. DELA protein will repress. DELA protein will come and repress all the functions of gibberellin. So functions of gibberellin like seed germination, stem elongation, flowering, fruit setting, internodal elongation will not be seen if DELA protein comes and bind. This is the whole story. And the one thing we should remember is soluble nuclear receptor protein. That is GID1. Gibberellic acid insensitive. And what they are DELA protein called in various other plants, in case of rice, in case of Arabidopsis. This is the short note about gibberellin signaling pathway. Hope this video is clear, and this video is useful for people who are preparing for CSIR, UGC NET Life Sciences and GATE Life Sciences exam. Thank you for watching this video. Thank you.